If you could please find your seats, we'll get started. Well, I hope you all had a wonderful evening last night. We really enjoyed hearing those stories and many more stories that we'll all share on Capitol Hill on Tuesday. We have an exciting panel this morning, but just wanted to pay attention to the schedule the whole day. We sort of switched things up at this year's conference to really highlight all the great work that the ALS Association's research department has been doing under the guide of Dr. Lucy Bruin. And we have many sessions today, so I hope you'll take advantage of all of them. Our first session is to talk about our federal programs that we'll be advocating for on Capitol Hill on Tuesday. So you'll have an understanding of what they have done, what they will do, and a chance for you to ask any questions of these program directors. Before I introduce my colleague, I just want to make one announcement. If you have not turned in your congressional meetings to the congressional meeting desk, please do so at the break of the session. We're trying to compile all the lists and make sure that if there are any early morning meetings, we have transportation for you and that we are able to get people who have late meetings as well. So next, I want to introduce my colleague and friend, Dr. Lucy Bruin.
So welcome everyone to, I hope, what will be a very stimulating session for you today. I wanted to right up front say that often we have a very large panel of speakers, and I recognize that it's sometimes challenging to get through such a volume of information. So we have focused today on two very specific program areas, which I think will be very exciting to hear about in depth. But also want to recognize that we work very strongly with many partners, NIH in particular and NINDS, and so although they have not representation today on the panel in terms of restriction of time and the value of having many other programs today, I just want you to know up front that we are very engaged with all our government partners and they play an integral role to our programs. So before, so I just have to get my... Before going into the session today and having my colleagues, Dr. Horton and Dr. Forsyth, speak about the programs, I thought it would be good to put a little bit of context in terms of how this fits in with our internal ALS research program. And I think, as many of you have seen the slide from time to time, we borrow this concept from industry, always talks about a pipeline. What our Treat ALS program is focused to do is really build a pipeline of research and bring early targets all the way through, support various areas of preclinical development, and have programs to help seed clinical trials and clinical management. Importantly, all of this has to be supported by model systems and biomarkers and tools that the community can use. One key example I use of how effective this program has been is the reality that SOD, a target that we identified in 1993, I know a long time ago, is one example where through partnerships with academia and industry researchers, we were able to support the very early funding and the FDA required IND enabling studies to move this now into clinical trials. But actually, I think a pipeline like this is a little misleading because by no means is it one straight road. It has many paths, but central to this is all of you living with the disease, and those of you who have lived with the disease through family members in the past. It's all about trying to identify the right targets to develop treatments. It's important once we've tested them, when they fail, to work out why it's failed and go back to the drawing board. And it's critical that all these components play together to finally get a treatment and cure for the disease. So our research program does many things, and one might argue it's very broad-based, but it's strategically broad-based. We fund globally in novel areas as well established areas in research, and actually we'll hear later today from Dr. Traynor the value of global research. We build consortia such as the Genetics Consortia, which we'll hear more about today, the Biomarker Consortia, and efforts around stem cells. We have a flexible program in that we can fund a good idea at any time of the year, and this has relevance especially as in last September when the breakthrough came for the new gene, which we'll hear more about today in Brian Trainor's session on chromosome 9. And we were able to very quickly mobilize the scientists in a strategic meeting and actually bring them together to identify key groups who could build model systems. I think what has been important, and I'll show a slide, is our support of the NEALS network to build an infrastructure and standardize activities in clinical trials. And most importantly, and I think relevant to our presentations today, is that we often are provide the initial seeding money for important studies that then can feed into government funding. Often it's young scientists who are just putting in for their first grants, or in other situations where there's been a really good study that the DoD program has funded, and there's now funding has ended, an opportunity to bridge fund and seed funding, and we have examples of that. And critically, over the years, we've built very strong academic industry partnerships through our research programs. And what it's all about, really, is bringing scientists and clinicians together worldwide to do a better job, identify the key areas that we should be researching, and often in those brainstorming sessions, think of novel ideas of how to tackle this very challenging problem. So unique to our programs is our fellowship program. We think it's critical to get new young scientists into the arena and commit them and engage them in ALS research. Our clinician scientist program enables young clinical fellows in a very challenging time where they have clinical demands to have protected time for clinical research in ALS. We have pilot studies which are critical to help some of the important studies of dose finding or biomarker finding for the larger clinical trials, which can cost as much as 25 million plus, but we can help seed and design those trials better. We have renewed our clinical managements program and intend to fund more in research today with those patients living with the disease. And we have examples where we already support interesting approaches to potentially look at whether compounds used for one particular indication might have some benefit for patients living with the disease now. And we have supported for the last, since 2007, the NEALS Northeast ALS Consortium network, which has really become one of the key networks to run many of the clinical trials within the US with its partners, WALS, and in fact has become a little bit of an example for developing networks in Europe. And part of this, and I know Dr. Bedluck is in the audience, and it was really his idea and initiative, and we now help support it through this program, is a Clinical Research Learning Institute, which really empowers patients to understand carefully about how clinical trials are designed, to come to the table and discuss issues. And I would highlight that we do have a clinical trial session today. I encourage you to attend, because if you have questions, we have many of the clinical trial investigators, the NeuralStem trial, Biogen's trial. Doug Kerr will be speaking about that, and Dr. Berry will give a super overview of everything that's going on. We encourage you to attend. So then just briefly to show specifically how our research programs can be critical for supportive activities. And when we learn more about the National Registry today, Laureen Nelson has been very pivotal in working with the registry to build the modules. And actually several years ago, she came to us to solicit funding for what she had as an idea, ACEs, an ALS Consortium for Epidemiological Studies. And critical to this was the idea that they needed to build consensus around collecting standard data, develop instruments for assessing risk factors, which has really been instrumental in helping build the registry, and use modular formats that investigators can then do their own studies worldwide. And this was helped by a website where researchers can collaborate. And in fact, this ACES Consortium has been pivotal in many of the epidemiological studies worldwide. And important is that if there is standardization, we can compare data from one area to another. And so this just demonstrates the many countries that have engaged and been involved in ACES to develop standardization in epidemiological studies. Another key area is and I don't know how much time Kevin will have time to address this, but the concept that potentially a biorepository could be a help to the registry and certainly is a need amongst the community. And the association has been committed for years to the area of biomarkers, critical for diagnosis, earlier diagnosis, and absolutely essential for clinical trials. And so a lot of the investment was initially to standardize how we get biomarkers. Just as we have to standardize epidemiological studies, we have to standardize how we collect data, how we collect samples, and how we share this worldwide. So this has been very critical to build a foundation, what might later be a repository with the National Registry, and has certainly been pivotal in collaborations for smaller studies. So then to the task of developing treatments and hopefully a cure for ALS. Not an easy task, and certainly one that takes many, many organizations, many, many partners. And so we're very, very pleased that the Department of Defense is focused on ALS in attempting to help us with this. It's a very slow process. Anyone from the industry will tell you that. I know that TDI is represented here and is doing also very good work and is finding it just the same challenges, that it takes time to identify the good targets and to bring it to fruition. Time, commitment, and funding. But what is also important is to have a better understanding of the disease, because it's only as good as whatever you put into the clinical trial that you'll get out of it. So in other words, if we don't understand how to treat the disease, what causes the disease, and what pathways are involved, it's very, very hard to get good treatments. And so we have been committed over the years to look carefully at the disease mechanisms and identify targets. And I think today one thing to say is that there is no one single approach, but many parallel approaches are very, very important to help us identify the disease, find the targets, and also many approaches to drug discovery. So key to this is really building partnerships, and so we value our partnerships with the CDC, with the DoD, with the NIH tremendously. Also our partnerships with many other ALS organizations and other disease organizations such as Huntington's disease. We learn from every aspect, and we really truly are in the same fight together to find treatments and a cure. What is so encouraging, I think, over the many years is to see the increased interest in the pharmaceutical sector. There are certain things that have made it that way. One is that there have been many challenges for the large pharmaceutical industry in even treating the blockbuster diseases like Alzheimer's disease. In fact, in communities of Alzheimer researchers, the big discussion is what happens if the current clinical trials for Alzheimer's fails. Many of the neuroscience sections in large pharmaceutical companies are closing. They are building very interesting new structures, and I think that we can play a big party to this. They are building many, many more academic industry alliances, so they don't do as much R&D, research and development in-house, but they look very closely to our academic colleagues to identify the targets and help bring them into a drug discovery forum. So it is no surprise then that Pfizer's, Bristol-Myers Squibb, large pharmaceutical companies see a real potential in the disease ALS. And that might come to you as a surprise, because many of you feel ALS market, not many people affected. Actually, if you all lived longer lives and healthier lives, there'll be a lot of people to treat. So we're very encouraged by the increased focus. We know that groups such as cystic fibrosis, who have a disease of similar size, have just been able to get a meaningful drug on the market. And obviously, as an investment, it's been a very significant one with their partners, Vertex. And I know that we can actually achieve the same if we work together in the community. It's also the small biotechs that play a critical role, because they help to build those ideas. And really what we do, and I think what the Department of Defense does tremendously now through the ALS research program, and we'll hear more about that, is really provide seed funds. The DoD provides quite a lot of funds compared to us, but it's an expensive enterprise. So if we can make the risk lower, then the likelihood of many, many approaches will succeed, and I'm convinced of it. So genetics is our tool in to finding the targets, and we have a great day lined up with first a good Bio 101 on human genetics, and we're very grateful that we will have that presentation. I think it will help many of you to understand the complexities of many presentations to follow. And we will hear from Dr. Trainer, who we're very, very proud to have and to know that he's made such an impact in the world of ALS research. And really the groundbreaking discovery of the chromosome 9 gene C9orf72 could not have happened without the bright minds of Dr. Trainer, Dr. Rosa Rademakers, and their many, many colleagues globally. So we're very, very pleased and excited about that. And as I said to many of you yesterday, the pie is being rapidly filled for familial ALS. And if we can believe what Brian says, and we won't hold him to it, but I'm hopeful that maybe within the next 5 years we will have solved all the genes linked to familial ALS. That doesn't give us the treatments yet, but it's the targets. We also focus on stem cell work, and you'll hear today in the clinical session some great presentations from the various clinical teams developing trials, and one of them, of course, is NeuralStem, and we were very critical in developing with funding Nick Boulis, the neurosurgeon in this trial, to develop what you see here as this metal frame, but not a trivial development to actually insert these cells into the spinal cord. And this development will be helpful in all the upcoming spinal cord injections and clinical trials, at least within the US, that are being planned. And then we work closely with NIH and my partners and colleagues, Amelie Gubitz and Margaret Sutherland at NINDS on the IPS Consortium. This is also a critical resource and tool for the community, and came from the finding that you can actually take skin cells from patients and generate from these the cells in the brain that you care to look at, in this case motor neurons and astrocytes. And many advantages to this, not only circumventing the challenging ethical issues, which we spent many years debating about and having views on, but the important thing is that you can now take this from patients that either you know their genetics or you don't know their genetics, and ask how they behave differently when you take them out and put them in culture. So not necessarily thinking of these as a tool for therapy directly by a transplant system, but using them for drug discovery in trying to identify if you can model the disease more effectively in human cells from the patients that have the disease, and then test your various strategies for drug discovery. And it's encouraging to see many of these kinds of things coming in as applications to the Department of Defense for funding. Added to that, we have a colleague that we are supporting at the Gladstone Institute, who actually, at the time when I first met him, was most focused on Huntington's disease, but I recognized that he had an interesting tool that we could potentially use for ALS, and got him quite engaged and excited to focus on ALS. He has recently published with ALS colleagues about how he could use his technology to identify a model system in a dish using TDP43 cell lines from a human patient. And what this most unusual glass box, actually it's Perspex box, is a microscope on which he can put these cells and in live time take recordings of what happens in these cells. So although they are not humans by any means, and they're only one cell, you can start to ask questions over time, and what is it doing? And this is all highly automated, and his team have put this together. All tools that will be effective in helping us solve this challenge, and brilliant for drug discovery. The industry is working with the consortium to see how they might fit into this and see how they might translate these technologies into the industry. And then biomarkers, as I indicated, such a crucial thing, something that really changed the field of MS for drug discovery. Having a biomarker speeds up clinical trials. Now we wait for the outcome, survival being the measure. We do have other scores, but in most cases, we decide whether a compound has been effective if there's improved survival. If we could have a much more definitive marker that tells us there's something happening and changed because of the drug much earlier, we can do many more less expensive clinical trials, so absolutely critical. And I could spend a whole morning talking about biomarkers, which I won't, but I just highlight one particular study. There are many fabulous colleagues who have done good work, but this to me is intriguing, and it's something that has come from understanding the basic biology of the neuron. And don't have to understand too much of it, just to recognize that NFH is an important molecule in motor neurons, neurofilament heavy. That's all you need to know. It's part of the architecture of our cells that function properly. And what is interesting, if you look at the image, and I can't be sure where to point, but you look on either side, you'll see on your left, control, those people that do not have the disease, low levels of NFH. In ALS patients, variable, because we don't all have the same looking disease, but certainly a huge trend towards an increase. Just an example of what a biomarker might mean, and I think this has huge promise and potential. And just shown differently, if you look over time in patients, and this is work done by Kevin Boylan and partnerships with Dave Borschelt and Jerry Shaw, we look in patients and we see the plasma levels are always high as compared to control. Variable again, because we have a variable disease, but in principle, very different. And then finally to note that clinical trials, as all of you in the room are passionate to be engaged and frustrated about because you might not be engaged in, so we have a whole session for you to ask the good questions. But a lot of this is dependent on good, solid studies for us to find treatments. And I'm very pleased to be partnering with Niels through our TREAT program, providing them infrastructure and support, which we started in 2007. And I'm sorry, the 2007 bar has kind of disappeared. There were trials before we came on board. We don't take all the credit. But it certainly, we do have a part to play. And I think it's a very important part, which we continue to do. So with that, I wanted to really just set the stage. I know it's a very whirlwind brief tour. Do come to the other sessions. You'll hear snippets here and there. But it's never possible to tell you all of what's exciting and going on. So I ask every one of you and encourage you, please come to our webinars. It's an hour once a month, and there's always a great scientist. They do their very best to tell the story in the most simplistic way. These aren't simplistic stories, but they certainly do their very best. Sometimes we've had challenges that we haven't had slides, and they've done a pretty good job. But in general, they run extremely professionally and very well. And we look forward to having you on our next one, where Virginia Lee is going to talk and tell us the interesting complexities of ALS and the spectrum of ALS and frontal temporal dementia. So with that, and I hope not to take too much more time, but I know it might set the stage for two wonderful presentations, and I welcome my colleagues. Dr. Horton will now give us an update on the registry. Thank you.
Okay, thank you, Lucy. And I just want to thank the ALS Association for inviting us here. We are always happy to come and talk to this group. Oftentimes government scientists, we can get really busy with a lot of different things, but it puts it into perspective when we come to conferences and meetings like this, and we can look out and really see people who are engaged. Last night was my first night at the candlelight vigil. Very moving. And this is why we do it. So this is why we're passionate, and very glad to be here today. So today I'm just going to give you a little bit of an overview of the National ALS Registry. I realize that some of you may have seen part of these slides. I apologize for that, but there are some of you, I'm sure, that don't know a whole lot about the registry, and my goal today is to make sure when you leave this room, you know what the registry is about, what we're trying to do, and some of the new initiatives that we're undertaking. So let me just preface this by saying we are the Agency for Toxic Substances and Disease Registry. We're a federal agency, and we're part of the Centers for Disease Control and Prevention, and we're located in Atlanta, Georgia, and the National ALS Registry is housed at ATSDR. So let me set the stage here. Back in 2008, the ALS Registry Act was passed, and many of you were instrumental in helping get this law passed. And for that, we at CDC ATSDR thank you, because that set us on our course to create and maintain the National ALS Registry. But I want to be clear, what it doesn't do is make ALS a mandatorily reportable disease. and that's important. So if you think about other diseases such as cancer, which in most, if not all states, cancer is reportable by law. That's why we know so much about cancer. That's why you can go and you know what the top cancers are. It's largely in part because this is a reportable disease. ALS, thus far, is not a reportable disease. But let me say that some of the methodologies that we're using with the National ALS Registry really helps us determine how common of a disease is ALS. So if you go and you look at the bill or the actual act, and this is on the CDC web portal, you can read the act. Some of the things specific in the act that the purposes for the registry are to describe how common is this disease from an incidence standpoint and a prevalence standpoint. Also, who gets the disease? How does it affect Caucasians versus African Americans versus Latinos? We don't really know that right now, but we're moving in that direction, and soon we hope to know how it affects different subpopulations. And then perhaps the most important part, and the reason we're all here, is why do people get ALS? We have theories and hypotheses, but we don't know for sure. So I'll talk about in detail how we're trying to answer some of these questions as we move forward. So many of you know the registry was officially launched in October of 2010. We had a huge fanfare of patients coming in and enrolling, and for that we're grateful. Patients are continuing to come in on a daily basis and enroll on a daily basis, and this is what we really need patients to do. In order to get the most out of this registry, we need everyone with ALS to come in and enroll. So this is one of the takeaways. I'll be mentioning that term a couple of times, but I want to make sure that people are clear on how we're actually capturing cases, or actually how we're populating our registry. So the first approach that we're using, and these are novel approaches. There are no other registries out there that we know of that are taking this approach. The first approach is to use what's already there. And by that I mean existing databases that people are familiar with. Medicare, Medicaid databases, VA databases. Just having access alone to these databases gives us a lot of information. And really what we do is we've developed an algorithm that we can run against these databases to actually tease out those that we feel have true ALS. As I mentioned, no other registry out there is doing this. And let me just, without getting too technical, mention some of the ingredients that go into this algorithm. So as you can see up here, some of the things mentioned, Realitech. That's a pretty telltale sign that someone has ALS. So that's built into the algorithm. Now I realize not every person with ALS is taking Realitech, but it's a start. Another thing, and this is probably one of the more critical ones, is the Icd Code. And this is pretty much a diagnostic or a billing code that physicians use. So we hone in on the one that's specific to ALS. Again, another good indication that this person has ALS if they're given this diagnostic code and it shows up multiple times in their records. And then other things we look at are how frequently does a person go to a neurologist? Is it one time? Is it multiple times a year? So it's not just one thing that we're using. We're rolling a bunch of different ingredients into this algorithm, and then we can apply it to these national databases. And I'll show you some numbers from our findings in a few minutes. And then the other approach, which I think most of you are familiar with, is our self-enrollment web portal. This captures patients. The reason we did this is this will capture patients who don't otherwise show up in the Medicare, Medicaid databases, the VA databases. And a good example is if you're not a veteran, well, guess what? You're not gonna show up in the veteran databases. So we felt we had to come up with some kind of safety net. How do we capture those people who may not be in these other databases. And regardless if a person thinks he or she is in the national or in the existing databases, we're encouraging everyone with ALS to come to the CDC web portal and enroll. And we do this for a number of different reasons. The first reason is the Medicare and Medicaid databases and VA, to a certain degree, there's a lag time. So what does that mean? By the time we get the data at CDC, that data could be a couple years old. So essentially, we're at the mercy of other agencies, and we want to speed that up. So if a person with ALS comes in and enrolls today, we know about that person today. So it's much more timely. The other really important thing about the registry is we have brief risk factor surveys, which I will go over. But this gets back to the etiology. What causes ALS? And the more people, the more patients who could come in and take these surveys, and let me mention these are brief surveys, the better our data will be, the more robust our data will be. So I can't overemphasize that enough. So this is just a diagram of what I've told you, or what I've just described. So the way this works, on the left-hand side of your screen up here is the national database approach. So after we apply our algorithm, it sorts people into one of three different categories. So clearly a person is not an ALS patient. Obviously, they don't go into our registry. That middle box there